Chapter 27, Introduction to Hematologic Disorders. So we'll go over just the basics of all the different hematologic neoplasms that we'll be covering, how often they occur, who it occurs in, why it happens, and how we can diagnose them. Well, we classify them according to FAB or WHO, and what are the molecular things that we can find, and the possible therapies that can happen with each. So generally, leukocyte neoplasms, we're talking about leukemias right now, not lymphomas. So with leukemias, one of the first things that we found out was with chronic myelocytic leukemia, was we found out that most of the neoplasms and malignant stuff is due to some molecular problem. Is some kind of chromosome abnormality. And the first one that was identified, which was 1960, was for CML, which is chronic myelocytic leukemia. That was the first one, and it was actually called the Philadelphia chromosome. So that sort of changed a lot on how we tried to identify and treat leukemias. So leukemias are broken down into the two general types, lymphoid and myeloid. Myeloid will include the myelocytic series, the monocytic series, and then we break them down into either they're an acute or chronic, which we'll explain with each chapter. Lymphomas are cancers that are seen in the lymphatic system. They're usually a solid tumor, like in lymph nodes, spleen, that kind of thing. Myelodysplastic syndromes is we're starting to see abnormalities, but it does not follow the criteria of considered to be a leukemia or lymphoma yet. So when we look at how we identify them, they use what's called the SEER technology, which is breaking down to surveillance, epidemiology, end results, and statistical analysis and data interpretation. This was actually developed by the National Cancer Institute, and they look at every single leukemia and lymphoma there is. All of this is put into NCI's database and is updated constantly so that we can come up with new protocols, which are treatments, how we identify the leukemia or lymphoma, and how we treat it and what kind of lab results we see. So in generalities, acute lymphocytic leukemia, ALL, is usually seen in young children. CLL, chronic lymphocytic leukemia, and MDS, myelodysplastic syndromes, are usually seen in older adults. And that we find out that most leukemias and lymphomas are due to, majority of the time, from either environmental toxins or exposures to them, some kind of virus or some kind of mutation in the DNA of the patient. So there's two different classification schemes that we'll go over that you need to know. The first one is FAB, which is the French, American, British. This actually started in the 1970s. And this one is totally based on morphology, us identifying the cells when we get the peripheral smear and looking at the bone marrow. We still do this FAB classification because for a while, this is all we have until we get results back from flow cytometry, the immunophenotyping, molecular cytogenetics, and all that to give us a better classification. So we have to look at the staining to determine if it's lymphoid or myeloid, and we'll go through all the FAB categories when we get to that chapter. The WHO, the World Health Organization, are constantly updating their classifications. We do have the most recent right now is 2022. And what this is based on is using immunophenotyping, flow cytometry, cytogenetics, molecular, in addition to all the morphological. So here we're looking at the lineage of all the cells, what the laboratory results tell us, so we can get a more pinpoint category. Because they start with the broad categories and break them into smaller things. If it's lymphoid, myeloid, or what. And then there's the international consensus classification, which the WHO falls in guidelines with. So one of the big things we see with a lot of leukemias, lymphomas is molecular pathogenesis. So what is happening is we're getting a DNA mutation. DNA is in the nucleus of the cell. For some reason, there is a mutation, and it's affecting hematopoiesis for the patient. So with that, we're getting dysregulation. We're getting problems with how cells progress through their maturation, or they actually stop, or it will actually inhibit certain pathways. So some of the mutations we see is due to the chromosomes. The chromosomes do translocations, they rearrange things. Sometimes we gain or sometimes we lose chromosomes. Sometimes certain part of the chromosome or the total chromosome is deleted. There are certain mutations in certain points in the DNA, or there can be insertions into the DNA sequence. Sometimes there's duplications of stuff. So we have to look at all that. So leukomyogenesis is the actual beginning of the leukemia and how it maintains it. And that's what we're trying to do by identifying what it is. So with molecular pathogenesis, like we just mentioned, was mutations and deletions. And with that causing the hematological malignancies, this will make us have uncontrolled proliferation of cells. They will just, instead of replicating in just, a normal day, 24 hours, 48 hours, they can actually replicate in as little as 4 hours. So cells can grow like crazy. And also, they don't die. The apoptosis is inhibited now. So they're more considered immortal. They live forever. And also, they can't repair the problems that they see in the DNA. With that, then we get problems with the cells maturing into their different cell lineages through the maturation sequence. So the differentiation of cells is blocked now. So we're seeing odd cells coming out. So there's epigenetic mechanisms that we will see where the DNA sequence itself is not truly altered, but there are inheritable abnormalities on how the gene is expressed. And with that, we can have problems with methyl groups that we see inside of the gene causing inappropriate expression of the gene themselves. And you can see the mutations. I mean, we're not going to really get into all this, but the mutations of all the nucleotides, the sequence of it can cause problems. With all that, usually what will happen is we have genes that are tumor makers. We call them oncogenes. And a majority of these are considered retroviruses. One of the first ones we ever identified, which we had already mentioned a few minutes ago, was the Philadelphia chromosome, which is seen in CML. And this chromosome is considered a proto-oncogene. In other words, with that there, it eventually will transform. It takes a little bit, but will happen. So it is a translocation of 922 of those two chromosomes. And with that translocation problem, we get what's called a breakpoint cluster region, ABLE. And what will happen is the cells do not die. They just live and they replicate quickly. Other oncogenes that we run into, and we'll be talking about this a lot, is called the JAK2, which is a kinase kind that causes polycythemia vera. It's seen in others, but the most common is polycythemia vera. Then there's FL23, ITD, which is seen in acute myelogenous leukemia. And again, we'll talk to this when we get to that chapter. So another problem that we run into molecular-wise is there are tumor suppressor genes. In other words, there are things there to stop cancer from growing. They help slow down the cell division. They help make sure that the cell does die when it's supposed to die. And cancers a lot of times will suppress this gene, and it's called TP53. So a lot of problems we are finding out now, which the WHO classification schemes look at, is this molecular issue. So there's DNA repair genes that are supposed to take care of these things. But when we start getting these oncogenes, then we start getting problems. We get a lot of mutations occurring, resulting in hematologically, leukemias and lymphomas. So we have different kinds of therapy. We're just going to go over basic therapy here right now. Is chemotherapy, which I'm sure you've all heard of. And this is giving some kind of chemical that's going to affect the cell in some kind of way. Either block the DNA from replicating or inhibit it from accepting folate or B12 or something to that nature. So there's different types of chemotherapy that can be given. Radiation is another one that can be done. That's very big in lymphomas when we have solid tumors. But radiation can also be given to a patient that's going to have a stem cell transplant, because they will radiate the entire bone marrow, kill everything, and then infuse stem cells into the patient so it can receive the bone marrow. Other therapies they have is supportive therapy, colony stimulating factors. In other words, these are the little things that are given to make the cells mature, like the granulocyte colony stimulating factor that can be done that makes the stem cells go to produce more of the myelocytic series. Like we can give patients injections of Neupogen or Neulasta, which is a granulocyte colony stimulating factor. So that can make us produce more neutrophils if we need be. But you have to be careful with that, because if somebody does have a myelocytic leukemia, that will make it worse. So the supportive therapy is done if their white count drops really low, chance of infection is there, we can give them shots of that and it will increase the neutrophil count to prevent infections. So there are also targeted therapies that we have. There's specific drugs for like CML, chronic myelocytic leukemia, and when we get to those leukemias, we'll talk about those targeted therapies. So the different types of hematological malignancies, statistically, what is the most common age for like ALL, CLL, MDS? What are the different causes? What is an oncogene? And just the basics of treatment. And we'll get into more depth when we get to each individual chapter. So just look at the chapter in your textbook, look at the questions at the end, and if you have any questions, please let me know.